Okay, good morning, everybody. And I'm going to continue what we were doing yesterday with ATP synthase. And so without wasting any further time, let's go straight to the lecture. All right, so this is what we had seen yesterday with regards to the structure of ATP synthase. And what we have found is that the ATP synthase, although it's a multi-subunit, it's a huge multi-subunit complex, it is kind of conserved through evolution. So if you take an ATP synthase in a bacteria, or if you take an ATP synthase in mitochondria of higher eukaryotes, you see that they are very similar in their structure and also very similar in the proteins that constitute these macromolecular complexes. So just by looking at this picture, you can understand what the similarities are, that there is some kind of a structure which they are calling here as the ring, C ring, which indicates that this would be a complex that is more like a ring structure. So this C ring is present in all of these ATP synthase that you may pick up. And then you have this kind of a head-like structure like the mushroom head. And it is composed of these alpha, beta subunits. And there are 3 alpha, 3 beta subunits. So it's a six polypeptide chain complex. And that is also present uniformly between bacteria to, say, higher eukaryotes. It could be even the chloroplasts. So all of them would have the same kind of configuration. And you see that between the C ring and this mushroom head, you have some polypeptides that connect them, that would form the stalk. So that when the C ring rotates, this rotation actually translates into rotation of the stalk, which then provides the conformational changes associated with the ATP synthase activity of the beta subunit. So it's always the beta subunit that is synthesizing ATP. The alpha subunit has nothing to do with it. The alpha subunit is there more like a regulator of this ATP synthase activity. So this is a catalytic activity, right? So alpha subunit, and this is a very recent study which has shown that alpha subunit can somehow regulate the ATP synthase activity of the beta subunit. So if the ATP concentrations are very low, then the ATP synthase activity of beta subunit is different from when the ATP concentrations are very high, which is actually the case inside the cell where you have a lot of ADP, PI as well as ATP. The concentrations are very high. But if you reduce those concentrations, if you reduce the levels of ADP, PI, if you reduce the levels of ATP, then the activity would be different, and that is regulated by the alpha subunit. It's a very recent study. I'm not going to go into much details of it, but from this very thing, you can understand that a lot of these structural enzymatic studies are very recent, and there is a lot that is still not understood about this entire complex. So then you see something that is stained in blue, this B prime, and the A that is attached to the C ring as well as to this mushroom head. So what this does is it acts. So presently it's believed that there is like at least threefold activity which I can mention. First is that whatever changes are occurring here is somehow translated to changes here. Whatever changes are occurring here is somehow translated to changes here. So any rotation here is somehow translated to some conformational changes in the head unit. The second activity is that if this rotor is continuously rotating, then along with the stalk, the head is also connected. But we do not want the head to keep rotating. Rather, we would like the head to catalyze ATP synthesis. So the head is sort of kept in check while the rotor and the associated stalk is rotating. And the head is actually kept in check by this associated B prime, and this delta or this OSCP unit, which sits on top of it and prevents it from undergoing something which we can call futile rotation. And the third activity is proton pumping. So, not pumping, pump is again, it's a word associated with active ATP hydrolysis. Rather, I should say proton diffusion, diffusion from a higher proton gradient to a lower proton gradient. So, what we saw was that there is higher proton gradient outside the cell if it is a bacteria, right? Ratnesh, outside the cell, bahar zyada proton. Or if it is in the mitochondria, then the mitochondrial intermembrane space has a lot more protons than in the mitochondrial matrix. And so the protons are going to be actively moving in, right? They're actively going to be moving in, and that is going to be turning this C ring. And the proton channels that are formed, part of the proton channel is also formed by this A subunit. This A subunit. So this entire complex has a lot of functions. Okay, this is something we saw the last time, and this is actually a model which was proposed by Paul Boyer in 1990s to describe how ATP synthesis might be done by the three beta subunits in the F1 particle, in the F1 particle, this particle. And what he proposed was that the conformational changes that are associated with the rotation of the central stalk. So the central stalk, this delta that he has shown here, is this delta. This delta. Oh, sorry, gamma. Delta. Delta. क्या दिखाया था? Gamma? Yeah, gamma. Sorry. Gamma. This gamma is this blue subunit which forms the stalk. So the rotation of this central stalk is translates into conformational changes of the associated beta subunits. And he tried to suggest that there are open conformations of the active site, which is actually catalyzing the formation of the phosphate bonds, or it could be a tight conformation, a loose, open, tight conformation. So so he suggested three such conformations. So you can see open, tight, and loose. So loose conformation is the conformation where an empty site is bound by an ADP and PI. So that is the first loose conformation. And then that moves into something called the tight conformation, which then catalyzes formation of ATP. So formation of that bond. And then once the ATP is formed, the ATP has to be released from this catalytic site. So there must be another change of this site that causes the formation of this open conformation, open state, where the ATP is released. So his model proposes three states: a loose state, a tight state, and an open state. An open state is when ATP can be released. A loose state is where in an empty pocket of active site, you have the ADP and PI go and bind. And a tight state is where it's such a conformation which is actively catalyzing the formation of these bonds so that ATP is produced. So ATP is bound to this tight state. So as this delta rotates, you can see that once there is binding of ADP and PI, then the ADP and PI is catalyzed into ATP, and then the ATP is released. So this is what Paul Boyer proposed. He proposed three possible states, configurational states in which the beta subunit can exist. From this diagram, do not confuse that this alpha beta subunits are actually rotating. They are not. They are rather fixed in space. It's only the motor and the associated stalk that is rotating, and the rotation is a counterclockwise rotation, so they are rotating, but the head is actually kept in place, as I have already suggested and I've said before, that this entire stalk kind of helps the head keep it in space and not allow it to undergo futile rotations along with this as the stalk rotates. Okay. Now this is the actual atomic model obtained from crystal structures of the beta subunit in closed conformation and beta subunit in open conformation. So this is what you're seeing here. You have the ATP bound here, and this is the ADP bound here, ADP plus PI, and this is the closed conformation or the tight state. And then you have the open state, and you can see the open state is favorable to releasing the ATP. So it allows the opening up of the active site so that the ATP can now escape out. So there indeed is a physical change in conformation corresponding to this. This was a hypothesis. This was a hypothesis in the 1990s. And in 2000, it is established that indeed you have these different states. And you can clearly see a state where the ADP PI is buried inside the molecule, and one state where the ATP can be released because the active site is now a little open, allowing the release of ATP. Okay, so here this is again a cartoonic depiction of the rotor. So far we talked about the head. So what we said is that you have this alpha, beta, alpha, beta, alpha, beta. So 6 subunits. And the beta subunit is the one that is binding to ADP plus PI, catalyzing ATP synthesis, and thereby it is able to produce ATP, right? That's what we have seen so far. And we have seen that, or we have understood that the alpha subunit that is associated with the beta also has a function, and the function is a kind of a regulatory function. And not a lot is known about the regulatory function, but a very recent paper does suggest that the alpha subunit is actually kind of regulating the ATP synthase activity of this complex. So at very low, so that what they did was they understood this through simulations of the reaction, as well as through doing experiments, that if you now subject this complex to very low levels of ADP plus PI, which is not present in nature. In cells, you really have high levels of ADP, PI, as well as ATP. And ATP levels are really kept high, and there's a very narrow band of this levels. So a little below this is actually very bad for the cells, and high above is also not right. So the ATP levels in cells, in any cell, bacterial cell, human cells, any cells, they're kept really high, but they're maintained within this very narrow range. So unlike these physiological concentrations of ADP, PI, ATP, if you lower it really low, then the energetics are really different, and that is controlled by the alpha subunit. इससे ज़्यादा मैं इस क्लास में और ज़्यादा डिस्कस नहीं करूँगी, because as I said, this entire regulation is a new thing. Not much is known about it. Okay. तो यहाँ तक तो हमने देखा है. अभी यहाँ पे आते हैं, इस पे फोकस करते हैं. तो हमें क्या पता है, ये एक मोटर है. This is a motor. So just like the motor we had seen in flagella, which was allowing diffusion of protons from outside the bacterial cell to inside, because outside there were much more protons than inside, and it was diffusion dependent, right? So in the similar fashion, this motor is also utilizing the proton gradients, the electrochemical gradients created by the proton gradients. So if it is a bacterial cell, we are saying that there is more protons outside compared to inside, just like we were talking about in case of flagella. If it is a mitochondria, we are saying that the F1 particle is facing the mitochondrial matrix, right? And so there, the FO, the fragment oligomycin, sensitive fragment, is the C ring, which is resident in the inner membrane of the mitochondria. and you have less protons in the matrix compared to the intermembrane space. So intermembrane space has more protons, and there is a proton gradient that has been established, and the protons now need to move along this gradient into the matrix. But the protons are charged, right? The protons are charged. Charged ions cannot move through lipid bilayer. So there must be a channel for their movement. So even now, we are not exactly aware of the exact path that is taken by these protons as they move through the rotor. So the rotor, we know a few key residues that are important, just like in flagella, जहाँ पे हमने देखा था, some residues, if you mutate, then the flagellar rotation is disturbed. Here too, we know that there are some key residues that help in this, but we do not know the exact path that it takes. We do know some key residues. So here we are going to discuss two such key residues that are important for the transport of the proton. Just two. There are a few more that are known, but as I mentioned, that pura channel ka pata nahi hai. We do not know the entire path. We do know a few residues. So I'm going to discuss just two residues. So if you remember in the case of flagella, do you remember which residue we had talked about as the mutation that could alter this proton movement? I'll just take a break. यह क्या मैंने पढ़ाया नहीं था क्या? Do you remember?
Ma'am, it should be more AB complex.
हाँ, more AB complex तो है. Did I talk about the proton channel, or did I miss that? Let me look at it myself. Did I miss it?
No, ma'am.
Oh, I did not talk about it at all. Bacterial motion. 1 second. All right. So I'm sorry, I missed that point. I will bring it up again next in the next class. So okay. अभी यही वाला हम देखते हैं. Are you able to see the slide that I have shared? This slide, the F0 electric motor. No.
No, ma'am.
Okay. I'll reshare it. Okay. So बात बहुत सिंपल है, और ये हम देखने के बाद हम एक्चुअली दो वीडियोस देखेंगे जिससे आपका आइडिया और थोड़ा क्लियर होगा, क्योंकि आजकल जितने भी ये बड़े लैब्स है जो इन चीज़ों पे काम कर रहे हैं, स्पेशली हु आर वर्किंग विद दीज ह्यूज मैक्रोमोलिकुलर कॉम्प्लेक्सेस, व्हाट दे डू इज दे डिजाइन वेरी beautiful detailed videos that allow you to visualize the movement as it occurs. तो बट उस वीडियो को देखने से पहले थोड़ा हम समझ लेते हैं कि ये चीज क्या है. तो आपके पास एक मोटर है और हम कह रहे हैं कि एक प्रोटॉन के मूवमेंट की वजह से ये रोटर ऐसे रोटेट कर रहा है. तो डेफिनेटली देयर मस्ट बी सम अमाइनो एसिड साइड चेन रेसिड्यूज that are sort of binding to this proton and transferring the proton, say, from one side chain residue to the other क्योंकि when we are talking about channels, it does mean that at some point you need to have some association of the side chains of the channels with these ions. So you're looking for some key residues. उन रेसिड्यूज को अगर म्यूटेट कर दे तो ये चैनल काम नहीं करेगा. तो यहाँ पे देखा गया है कि ये जो रिंग है, जो रिंग लाइक स्ट्रक्चर बनता है, either it could be 14 chain ring, 10 chain ring, 12 chain ring, depends on the on the organism, depends on where it is, mitochondria का है, bacteria का है. तो ये जो ring like structure है, each of these subunits will have one key residue that was identified as an aspartate. So aspartic acid. And flagella में भी एक aspartate residue है, which is important. If you mutate that aspartate residue, then you would see that the proton gradient is actually not easy. The proton transport is not is I've forgotten, it's either blocked or maybe partially blocked. So वहां पे भी एक aspartate residue था. Now aspartic acid का charge क्या है? साइड चेन चार्ज क्या है?
मैम नेगेटिव चार्ज.
हाँ रत्नेश, aspartic acid में side chain charge is negative because of the presence of a COO minus. COH group है end में, तो इसकी वजह से it has a negative charge. So you can now understand why it would be something that would like to bind to a proton. अगर COO minus है, it would like to bind to a proton H plus. So this aspartate, just like in ATP synthase के C ring में जैसे एक aspartate है, similarly ऐसा एक aspartate flagella में भी है. So it's the same analogy. In both these cases, there is an aspartic acid residue which is responsible for binding this proton. So just binding a proton is not enough. As I said, the proton has to move through the C ring, right? That means it has to have different points where it would bind, then it would be transferred from this residue. इसकी movement होने चाहिए. So what is shown in this picture are two key residues. एक तो है aspartate या aspartic acid जो आपके C ring के पॉलीपेप्टाइड चेन के साइड चेन पे है. अभी ये जो aspartic acid है, this aspartic acid is residing in the membrane, inside the membrane. ये पूरा जो आप देख रहे हैं, सोचिए कि इसके आसपास सारा कुछ phospholipids है. So the phospholipids are non-polar tails, right? They have non-polar tails. They would not like to see a charge here. COO minus is not something that is energetically favorable in a highly non-polar environment of the lipid tails. So what happens is this COO minus tends to be buried inside until you have a proton associated with it. That proton association with it allows it to move a little, थोड़ा सा उसको open कर देता है. And so what another thing that happens is the presence of this A. ये जो A subunit यहाँ पे आपको दिख रहा है, which is part of this complex that keeps the C ring attached to the head of the mushroom. This A is also part of the channel, the proton channel. And there is one residue here which is an arginine residue. So this arginine residue, see, it seems that as if the proton binds to this arginine residue, यहाँ से वो इस aspartate के ऊपर चला जाता है. Then there is a slight movement of this ring, and in the process it is turned back into it is moved, that proton now moves further along the channel. So वो यहाँ से निकल जाता है. So then there is another proton that comes, binds to arginine. Arginine hands it over to the other aspartate residue and moves forward. It's a little difficult to comprehend in this way, but just for your information, there is at least two residues I'm trying to highlight here. One aspartic acid residue and one arginine residue. And स्पेसिफिकली आप याद रखिए कि एक एस्पार्टिक एसिड रेसिड्यू है जो फ्लैजुलर मूवमेंट में भी इम्पोर्टेंट है और एटीपी सिंथेस मूवमेंट के लिए भी इम्पोर्टेंट है. बोथ ऑफ देम आर एक्टिंग एज पार्ट ऑफ द चैनल. जो पूरा चैनल है, द पाथवे द प्रोटॉन्स टेक टू मूव फ्रॉम दिस हायर कॉन्सेंट्रेशन टू दिस लोअर कॉन्सेंट्रेशन रीजन. जो चैनल लेता है this proton, it's part of that channel. And if you mutate this aspartate residue, then the proton movement is either completely blocked or partially hindered. without saying anything more, what I will do is I will run this video. As you can see, this is part of the Stoke lab, and it's a beautiful simulation, almost an exact simulation of what ATP synthase function is known so far in literature. So let's have a look at it. Okay. Uh, uh, Let me play this video and please let me know if you can hear the. So are you able to hear what is being said along with the video? Is it audible to you? क्योंकि वीडियो के साथ एक ऑडियो है, वो आप सुन पा रहे हैं?
No, ma'am.
No. Okay.
No, ma'am.
So I will narrate what is being said in the video and just have a look at the video. So initially वो क्या बता रहे हैं आपको कि ATP का स्ट्रक्चर बताएंगे. ये this is kind of a space filling model, and what you see here is ADP, and they will add, or ATP. No, this is ATP. I think this is ATP, and you will see how they show. So यहाँ पे वो बता रहे हैं कि there is ATP and there are 3 phosphate groups, and this is the structure of ADP and this is PI. So what ATP synthase does is it catalyzes the formation of ATP from ADP and PI. So you need to bring these two highly charged molecules together. So okay, यहाँ पे जो आप देख रहे हैं, this is the ATP synthase. अब तक हमने जो describe किया है, ये वही ATP synthase है. This is the membrane in which it is embedded. You have the C ring, right? As I said, 10 polypeptide chains हो सकते हैं, 12 हो सकते हैं, 14 हो सकते हैं, लेकिन ये ring like structure में होंगे. And you see this associated stalk. This part is the A subunit that we have seen before. This region would be part of the channel. You will see protons that would follow a part that is associated with A and then would be released in the matrix. If this is the mitochondria, then this would be the matrix, and this is the intermembrane space. If this is bacteria, ye outside the bacteria hoga, and this would be the cytoplasm. Okay, this is the 6 subunit head, consisting of 3 alpha, 3 beta, right? And you would see the ATP synthesis occurring here. This is the stalk that would channel the movement of this C ring to conformational changes in the beta subunit that would catalyze ATP synthesis. So let's look. So in this video, what they're describing is a mitochondrial ATP synthase. And you see these things, these yellow dots that are jumping around here, these are the protons. And you see there will be less of these protons here and more protons here. Now ADP and PI, you see, is already now comes ADP is bound to the beta subunit. It catalyzes ATP synthesis and it is released. So this is okay. So here they are describing it as factor 1. And here that you see there is a channel formed between the C ring and the A subunit, which is allowing the movement of the protons. And we have seen that at least 2 subunits, 2 side chains are important. One aspartate, which is present in this C ring, and one arginine here that is helping in the transport of this proton across the channel. Now this movement of the motor is connected to the movement of the central shaft. Okay, that's how do I go back on the video? One second. It's very difficult. Maybe I should. I'm going to stop this. I'm going to try to play the video separately, so that I have control over the video. Um, just give me a second, please. Okay, so we come here. This is the structure we were seeing so far, right? We said that there is a channel that is formed between this motor and this A subunit, which is allowing the movement of the motor. So every time the proton binds and then is released, it causes a small change in the angle of the motor, a small torque. And we saw last time that the torque is something about uh, oh, no, I'm confusing it. The torque of about 14 degrees was for flagella, or the torque for 14 degrees was for ATP synthase? I think it was for flagella. Anyway, so this causes a small change in angle, a small torque every time the proton binds and is released. So binds from the surface, from the intermembrane space, and is released in the matrix. Every one such movement causes 1 degree change, or a change in the angle of rotation of this motor, and that translates into rotation of the stalk. But just keep notice of the fact that this head is actually not rotating. Neither is the associated B beta prime. So what happens when the stalk rotates? Every time the stalk rotates, it translates into some conformational changes that are occurring in the beta subunit. The associated beta subunit is not rotating, neither is the alpha. So the head is in space, intact in space. It's in position. The only thing that's happening is that there is a conformational change associated with rotation of the stalk that catalyzes the formation of the phosphate bonds between ADP and PI, and also creates the open conformation that helps in release of ATP. Because what we have learned is there is an open state, a tight state, and a loose state, right? A loose state where ADP and PI come and bind, a tight state where the catalysis of ATP formation takes place, and an open state where the ATP is released. Let's go back. So you see that this is the central stalk that was represented as gamma mm, uh, in Paul Boyer's description. And let's look at it once more. So here an um, you have an loose state that goes into a tight conformation, and then there is an open conformation where the ATP is released. Loose state. Uh, let's look at it once more. Let's look at it once more. Let's follow one of the ADP and PI. So, okay, if we look at it, this is a loose state. Then there is just a conformational change, a tight state where ATP is formed. Now the ATP has to be released. It's an open state. Again, this is another loose state. Then again, this would become a tight state, and then an open state where ATP would be released. So all of this, whatever Paul Boyer had suggested, is actually taking place, and using um, extracrystallographic studies, it has been found that indeed these three states exist. And these three states exist because of the rotation of this associated gamma, or the shaft that is linking the motor or the C protein to this entire top complex. All right. So, okay. Now, this is another interesting thing that we are going to see here. In mitochondrial membrane, in the mitochondrial inner membrane, these ATP synthase complexes do not exist as uni complex. Rather, they exist as dimers. So two such complexes are known to come together the way that they have shown it here, exactly in the similar fashion. They are known to come together with the help of the associated these chains, and they are so the association is known to induce some amount of membrane deformation or membrane bending at these sites. And what is believed is that this creates a region in the mitochondria where you have a pocket that is very high, that is sort of highly synthesizing ATP, as well as it is creating a method by which mitochondrial membranes are actually highly looped. So if you think of mitochondrial membrane systems, mitochondria, how do we visualize the mitochondria? By thin sections that are seen under electron microscope, right? So if you look at a thin section, you will see that if this is a mitochondria, the inner membrane is actually highly looped. Remember that the inner membrane is highly looped. So what is believed is that these loopings are created, very heavily created by these ATP synthase molecules that dimerize, and the dimerization induces membrane deformation. Now this would give you an idea, at least when I had read this, to me it seemed कि ATP synthase फिर वहीं पे है जहाँ पे ऐसे membrane bend किया हुआ है, जैसे so if this is a sheet of bilayer membrane, think of it as a sheet of bilayer membrane, तो हम कह रहे हैं कि अगर दो डाइमर्स यहाँ पे अगर यहाँ पे दो ATP सिंथेज है, वो इस तरह से डाइमराइज कर रहे हैं that they are inducing local bending of the membrane. Local bending of the membrane. To me, when I was reading it, I thought कि अगर ऐसा एक inner membrane है, माइटोकॉन्ड्रियल इनर मेम्ब्रेन ऐसा है, तो एटीपी सिंथेज सिर्फ यहाँ पे है, मुझे ऐसा लगा था पढ़ने पे. But then I read further and I've realized that that's not the case. What they're saying is that there is a local bending, which means that you would also have ATP synthase all around this. हर जगह पे है, लेकिन क्या हो रहा है, एक local bending हो रहा है. So of course this bending is dependent on it, but if you have two such local bendings coming together, you would see that that region would also try to flatten out. So it's a little difficult to understand that way. So I just want to ये मैं इसलिए बता रही हूँ ताकि आप लोगों को भी ये कंफ्यूजन ना हो. इनर मेम्ब्रेन थ्रूआउट यू वुड सी एटीपी सिंथेज. ऐसा नहीं है कि जस्ट जहाँ पे इनर मेम्ब्रेन ऐसे बेंड किया हुआ है सिर्फ यहीं पे एटीपी सिंथेज है. आपको यहाँ भी दिखेगा, यहाँ भी दिखेगा. What is happening is that the dimerization is causing a local bending of the membrane. And okay, तो इस वीडियो को आई होप यू आर एबल टू सी द स्क्रीन. तो ये वाला हम फिर से देखते हैं. So what we see is that two such subunits can come together, interact with the help of these associated chains or polypeptides, and cause a local bending, and that creates an area where ATP is being very highly synthesized. It's like it's a region which is क्या कह सकते हैं इसको? How do I explain this? It's a region which is highly catalyzing ATP. It's like concentrating the entire thing in one small region of the entire mitochondria. Okay. Now I'm going to go back. I'm going to go.
Ma'am, I have a doubt
Yes, Sarika. Go ahead. While I share my screen, just go
Is
ahead.
this, is this local folding is not just due to a dimer, but a multiple of dimers, means a series of ATP say ATP synthase?
Yes, a series of ATP synthase, but what is now what ये सारे जो काम जो मैं आपको बता रही हूँ ये recent हुआ है. So initially the idea was exactly that I have said कि लोगों को ये पता था कि ये dimerization if the two molecules come together, then the angle at which they are coming together is causing a local deformation. So this much was known, but it was also known that all throughout, even the regions that look very straight under the microscope also have ATP synthase. So now the idea is that multiple such dimers are there in those regions, and the presence of multiple dimers is actually causing local deformation but also inhibiting very long range deformations. These are all sort of molecular studies. Like what, how do I explain this? It's more like simulation studies that have suggested how the deformation of membrane propagates along the mitochondrial inner membrane if you have if you now start to have multiple such dimers assembled one after the other. But um, अगर आप वो पेपर पढ़ना चाहते हैं मैं आपको वो लिंक भेज सकती हूँ. For the moment, it's enough for us, perhaps for this class, to understand that if you have dimerization, you have the propensity to create a local deformation of the membrane. And why this is preferred? It's a hypothesis. It's a hypothesis, not proven. It's a hypothesis that maybe having something like this creates a local high concentration region where this reaction is taking place. It's as if you have this entire mitochondrial matrix, and mitochondrial matrix में diffusion हो सकता है, right? So a lot of these particles are moving all around. But what if you want to have a reaction occurring at a very high rate? Then you would want all the members of this reaction to be at a very close range, very closely spaced, so that in a very so that things are not moving all around, rather in a very small space they're condensed. That would be more of the term. They're condensed into a very close space, so the reaction rates are very fast. One of the mechanism is that इसी membrane के ऊपर very short distances you find this respiratory complex 1 2 3 4, which are creating these gradients. Then you have these ATP synthase molecules, and all of them are very closely spaced so that the entire reaction has a very high rate of occurrence. So the rate of occurrence of this reaction, so ATP synthesis, uh, I, I think I read somewhere it's about 400 molecules of ATP that are synthesized per second by one complex itself. So for the system to achieve these high rates of ATP synthesis, it's perhaps a mechanism. Again, it's a hypothesis, not proven, but a hypothesis that it's perhaps a mechanism to keep everything in one place so that the reactions can occur very fast. Have I answered your question, or have I gone too far off your question?
No, ma'am, you have answered it.
ओके ओके सरिका. ये जो हम लोग पढ़ रहे हैं ना, जो जितना आप लोगों ने टेक्स्ट बुक में पढ़ा होगा शायद उतना उतना ही है. बस उससे ज्यादा हम लोग जो बात कर रहे हैं, those are things that are very recently found in literature and perhaps not yet in textbook. So what I'm trying to say is giving you a flavor of what is perhaps the recent knowledge, and some things which are not known वो भी मैं आप लोगों को बता रही हूँ. जैसे मैं बता रही हूँ कि प्रोटॉन चैनल्स, प्रोटॉन एग्जैक्टली किस पाथवे से जा रहा है ये पूरा हमें पता नहीं है. इट्स स्टिल अ मैटर ऑफ रिसर्च. लेकिन कुछ कुछ रेसिड्यूज हमें पता है. जैसे आप एस्पार्टेट को म्यूटेट कर देंगे तो फिर ये प्रोटॉन चैनल्स वर्क नहीं कर सकते. आर्जिनिन को म्यूटेट कर देंगे ऑफ द ए सब यूनिट वर्क नहीं कर सकता है. ऐसे कुछ और हमें पता है लेकिन पूरा चैनल नहीं पता. जैसे हमें ये नहीं पता है कि एग्जैक्टली ये मेम्ब्रेन डिफॉर्मेशंस मतलब एग्जैक्टली कैसे होता है. देयर आर सम स्टडीज which suggest membrane deformations might be getting nullified if you have too many of these dimers one after the other. But again, it's not it's not very well studied, not very well studied. Okay, oh. So what I will do now is, and this is the last thing that I would do. I had promised that we would look at how the, so what I had said is ATP synthesis के लिए the toughest part of ATP synthesis is that you have ADP which has these negative charges. You had inorganic phosphate which has a negative charge, highly negatively charged molecules है दोनों दोनों को एक साथ लाके जोड़ना है, which is energetically unfavorable. If you think of it, you want to bring two highly negatively charged molecules together is energetically unfavorable. एक positive होता negative होता would have been easy, right? Then how does this entire catalysis take place? तो मैंने कहा था कि इसमें एक और चीज है कि जो एक्टिव साइट है जहां पे एटीपी सिंथेसिस हो रहा है, that has side chains which are stabilizing this molecule, the ADP as well as PI. Along with this, there is magnesium ion, which is also helping to stabilize these charges, because otherwise these two highly negatively charged molecules wouldn't want to come together. If you remove magnesium ion from this reaction, the reaction would not proceed. So I had said we would look at some molecular structures to understand how this happens. And what I found is a beautiful video that actually shows this. So I'm going to play the video. What I will do is I will start sharing my screen. Uh, can you see my screen?
Yes, ma'am.
Yes, ma'am.
Okay. So again, this is the what this video is going to show you is again ATP synthase, but it's going to zoom into the active site. और एक्टिव साइट में आप देखेंगे कि एडीपी जब एक्टिव साइट में बैठा है, then how it is linked to the side chains of amino acids that are present around the active site. You will see how the phosphate molecule is brought near it. You would see how magnesium ion is stabilizing the charges on the ADP because it has highly negative charges. And in the end you will see how the phosphate has to be aligned with the phosphate groups on ADP so that it forms more of like a planar structure. It's all in on one single two-dimensional plane so that the catalysis can take place. If this planar conformation is not achieved, you wouldn't have formation of the bonds. So we will see that in the video. It's a beautiful video to explain this, the best way to do it. Are you able to listen to the associated audio for the video?
Yes, ma'am, but not clearly.
Not clearly. Okay, let me try. Let me try. Maximum. What I can do is I will switch off the audio and play the video once more so that I can voice over and you can understand the video. 1 second. So what you're seeing here is, so again, this is the ATP synthase. You see the F1. You can see the shaft rotating because you're looking above the membrane. Inside the membrane, you have the C ring which is rotating. You have the alpha, the beta subunit, and we know the beta subunit is catalyzing ATP formation, right? The formation of ATP is occurring in the beta subunit. Now, the rotation of the motor, the anti-clockwise rotation of the motor is seen to rotate the shaft, and then that is catalyzing conformational changes of the beta subunit. ये है एक beta subunit. This is called a ribbon structure, which gives you the secondary structure of the protein. This is ADP, 1-phosphate, 2-phosphate, and you see the ribose ring. And now you're seeing all the associated side chains which are stabilizing this ADP. Let's go back. Let's go back. Let's go back. Let's go back. So ये है beta subunit. इसके यहाँ पे आके ADP ने बाइंड किया हुआ है. It's binding to the phosphate and is released as ATP. So if you zoom in on this, you see all of these are all polypeptide chains. So there are side chains here which will be stabilizing this ADP. So here you're looking at side chain interactions. I'll just stop here. यहाँ पे आपको जो दिखाया जा रहा है, these are hydrogen bonding interactions which are occurring with nearby side chains. With nearby side chains, and with this ADP molecule that is placing the ADP molecule in space inside the active site. Because active site में अगर ADP ऐसे ऐसे हिलता रहा तो फिर आप catalyze नहीं कर सकते. You need to place it, you need to kind of hold it. Exactly what we have seen in optical tweezers. Optical tweezers में using the beads and using that highly collimated laser source, they were holding the actin fiber or holding the myosin fiber, right? Here you need to hold the ADP. The way it is held is with the help of these hydrogen bonding interactions that are occurring with the amino acids that are present along the polypeptide chain. उनके side chains के साथ hydrogen bonding interactions करके it's kind of holding this molecule in space. Okay. So you can see all the interactions that are occurring here. Now you should be able to see how the magnesium ion, this is the magnesium ion, and it is stabilizing the interactions that are occurring with this phosphate. So this was the adenine residue, this is the ribose, these two are the phosphates, and these had to be stabilized by the magnesium. Let's go back. These two phosphates, यहाँ पे और यहाँ पे, ये जो त्रिकोण जैसा आपको दिख रहा है, ये है आपका phosphate, and these were stabilized by the magnesium ion. Now you have the next phosphate group coming in. But when the next phosphate group comes in for catalysis, it has to be reoriented so that they're all in the same plane. If they're not in the same plane, this catalysis wouldn't take place. And this reorientation is also occurring with the help of the hydrogen bonding interactions that are taking place with nearby molecules. So the entire system is helping in catalyzing ATP, formation of ATP from ADP and PI. And it's happening within the active site of ATP synthase. And the active site is in the beta subunit. And the entire process is driven by conformational changes that switch from the open conformation where ADP and PI come and bind to it, to a tight conformation where the actual catalysis is occurring and you have ATP formation, to an open conformation where ATP is released. Now all of these conformational changes are occurring because you have a C ring that is allowing diffusion of protons along the concentration gradient, and the channel that is formed is part of it is also associated with this associated alpha A subunit, and you have the C ring, so you can see the protons that are moving through the channel. We know some amino acids that are part of it. We know that one aspartic acid residue is very important in this catalysis. And what the proton movement causes is it causes a counterclockwise rotation of this motor. Counterclockwise rotation of the motor. And the counterclockwise rotation of the motor is translated into a counterclockwise rotation of the associated stalk. So if we look at it from here, you would see that the rotation of the stalk is occurring because you have the rotation of the motor that is embedded inside the membrane. Inside the membrane, you have the motor that is rotating counterclockwise because it is powered by the movement of the protons along the concentration gradient, and ultimately all of this helps in the catalysis of ATP. So we stop here, and I will take any questions if you have. If you have any questions. No questions? All right, then what I would do is I will first stop recording.